Okay, hopefully this is getting to you guys. And I'll be checking kind of periodically throughout just to make sure things are streaming. So if you notice me kind of phase out for a second, I'm just checking that at least there's a feed being noticed online. There's also a delay between when I'm going to be kind of giving you lecture material versus when you guys actually see it. It might be a minute or two. So if you do type in a comment or something or a question, I'll do my best to kind of go back and deal with that, or even maintain or answer all the questions near the end of the lecture. So to be honest, I'm kind of nervous, so you might find that in my voice as we go along, just because I'm not sure how well this will work out. But let's hope. And if you can give me feedback at the end of the lecture, that would be great as well. Either by email or just, I don't know, that'd be great so that we know whether or not this is going to work on a regular basis. So the plan today then, once you guys are ready, is to go through a bit of metabolism. Again, reviewing what we saw last week before the exam, and then building on that. So for the most part, we've got, as I've said a number of times now, we've got the phosphagen system, glycolysis, and then finally we'll have the Krebs cycle, and then also the breakdown of free fatty acids through beta oxidation. In humans, for the most part, that's where most of your ATP comes from. There is some use of protein and amino acids, but this can usually makes up about 5% of what you actually use on a daily basis. So I'm going to just double check that the feed is working, and then we'll get down to business. So the plan. Okay, I just double checked all the audio, and so we'll get on to the actual lecture. Hopefully we'll see how it goes. So we'll just transition into the lecture slides now. And voila. So last class we left off just before this, so just give me a second. Just taking a look at different ways that ATP can be broken down in muscle. Specifically, we looked at ATP breakdown through hydrolysis and an ATPase. So those ATPases, which are in red on this particular slide here, those can be sodium-potassium pumps, they can be calcium pumps, or they might even be your myosin heavy chains, which are types of ATPases. Again, we get the production of a hydrogen, which is interesting from a metabolic acidosis perspective, which we'll talk about in a little bit. And also from we also get the breakdown products of the ATP and a fair bit of energy. And most of this energy, as I mentioned last time, is released in the form of heat. So this is a fairly inefficient process in that of the ATP that you produce, only about 30% is actually created from carbohydrate breakdown, and 70% of the carbohydrate that's broken down actually creates heat. So in general, you're pretty inefficient, even though the electron transport chain is the best system that's been figured out with genetic or evolution. The other reaction that we had was when two ADP molecules come together through the adenylate kinase reaction to produce ATP and an AMP. And interestingly, AMP is one of these molecules, a signaling molecule that actually induces a lot of changes in gene expression within a muscle. So when you have severe ATP depletion, that creates a lot of AMP, and AMP signals adaptation within a muscle, which is quite interesting. So the resynthesis side of things, as we mentioned last class, involves ATP being created through the breakdown of phosphocreatine, or creatine phosphate's another name for it. And you'll see that when you do this process, you also consume a hydrogen ion, which obviously increases the pH in the muscle as time goes on. So again, the muscle isn't very high in its ATP concentrations, and in fact, if you ran out of ATP within a muscle, really only one thing could happen. And what do you guys think that is? I'll give you a second, but I imagine I won't get the answer anytime soon. Right, you would have a skeletal muscle actually die. Anytime you deplete ATP to levels that are basically zero, that cell will die. And in the case of muscle, you'd have a muscle cell that would die. So all the time you have to have at least a basal amount of ATP there. Otherwise, the cell will actually stop contracting or stop undergoing, say, calcium movement in order to conserve the ATP and maintain the muscle alive. Now our next slide here, we get into glycolysis, specifically the first steps, again, anaerobic glycolysis, in other words, not requiring O2. And now there are a series of enzymes here that I'm not going to go into. The key points that we need to remember is that anaerobic glycolysis involves the breakdown of glucose. The first step is entry into glycolysis of a glucose molecule, which may arrive either from the bloodstream, and it crosses across the plasma membrane through a transporter that we talked about a few months ago, actually, called a GLUT4 transporter. So these GLUT4 transporters bring the glucose across the barrier, which then the glucose gets quickly phosphorylated. So you get the use of an ATP. And by actually consuming ATP, you quickly convert glucose into glucose 6-phosphate, and thus glucose within the cytosol is always maintained at a very low concentration, so that you can always have the movement of glucose across the membrane down its concentration gradient. The next couple of steps are just reorganization of the glucose molecule until you reach step number 3, where we see another ATP being used in glycolysis. This is an irreversible reaction. In other words, once glucose gets past this point, it can't be resynthesized. So there are a couple of points in glycolysis that are irreversible. Now you'll note at the bottom of this slide here, let me just put my arrow on the screen, that you have a dividing arrow. So essentially, at this fructose 1,2 biphosphate molecule, you still have a 6-carbon molecule. And once it goes through the next enzymatic reaction, it breaks down into two 3-carbon molecules. So all the steps beyond this slide are done twice. So moving on from fructose 1,6-biphosphate, you get the production of 2 3-carbon molecules that you can see here, eventually resulting in 2 identical molecules that will go through the last stages of anaerobic glycolysis. So those stages will include a few things. One, you'll have reduction of an NAD into NADH plus a hydrogen ion. So there is some production of hydrogen ions at this stage. This is these reduced substrates end up either entering the Krebs cycle or maybe used in the lactate dehydrogenase reaction that we'll see in a little while. Moving further on, you finally get your first 2 steps where ATP is actually produced in glycolysis. So here at step number 7, you have the first point where ATP is being produced. And because you had two 3-carbon molecules, so two molecules that are 3 carbon in length, you'll actually go through this process twice to produce 2 ATP at this stage. And then finally, in the last reaction, before you produce pyruvate, you also produce 2 additional ATP. So in total, you get 2 3-carbon molecules at the end of glycolysis. It's irreversible, so all these reactions can't be undone in a muscle cell. And so if you wanted to deal with the products of glycolysis and go back to producing or at least reconstructing glucose, you have to do that in a different cell in the body. So pyruvate, and essentially once it's converted to lactate, can be moved out of the cell and then taken up by other molecules, or excuse me, other tissues where it can be turned back into glucose. The other thing you get is 4 ATP being produced and 2 hydrogen ions. But the net result is that you also get, you also get reduction of 2 NAD that produces 2 NAD pluses, NADHs, excuse me, and 2 hydrogen ions. So in total, if you break down one glucose molecule, you'll end up with 2 ATP after you take into account the 2 ATP that you use to start the reactions. And then finally, if you were to use glycogen as your source of glucose 6-phosphate, you would skip the first reaction, which I'll just show you up here. Glycogen enters actually at glucose 6-phosphate. So you can kind of consider glycogen above this glucose 6-phosphate molecule and entering or adding a glucose 6-phosphate molecule whenever you break down a little bit of glycogen. So the end result, or net result, if you use glycogen as your source of glucose 6-phosphate, is 3 ATP. So hopefully that's fairly clear. I know you guys have all seen this before, and I just want you to remember again the important enzymes in that process, which were phosphofructokinase. It's most important because it is the one that needs to be activated the most to allow glucose molecules to be broken down quickly to produce ATP, which is your second fastest way of getting ATP in a skeletal muscle. But that doesn't come without consequences. So the final stages of glycolysis essentially are the production of pyruvate here that you'll notice. And with the production of pyruvate, you may get an accumulation of this substrate if the Krebs cycle, or the enzyme that enters pyruvate into the muscle, isn't fully activated. And that enzyme is pyruvate dehydrogenase. It is the rate-limiting enzyme bringing glucose, or essentially pyruvate molecules, into the Krebs cycle. So if you get an accumulation of pyruvate in a muscle, what happens is this reaction, the lactate dehydrogenase reaction happens in order to get rid of the pyruvate and to recycle the NAD. In other words, this reaction consumes NAD that was previously produced in glycolysis in order to maintain flux through glycolysis. Because if you had an accumulation of pyruvate at the end of glycolysis, it would essentially back up all the reactions that happened previous to it. And then the other thing that we should note is that there is some net production of hydrogen ions from glycolysis itself. And these hydrogen ions can induce acidosis, or a change in pH within the muscle, which can have dire consequences for contractile function. In other words, muscles won't be able to contract so well if they continue to accumulate these ions within the cytosol. So what does this mean? Does the production of lactate lead to metabolic acidosis? That's the question that is on everybody's mind. So the actual reaction that creates lactate, as we saw last class, consumes hydrogen ions. So if we look in this, this diagram, we have pyruvate combining or interacting with NADH. And in the process, there's another additional hydrogen ion that gets taken up and popped onto the side of what was originally a pyruvate molecule, but is now considered a lactate molecule, as it now has an extra hydrogen on this side, as well as another hydrogen being donated from NADH. So the reaction that produces lactate and regenerates NAD consumes hydrogen ions, which means if you consume hydrogen ions, you're taking them out of the cytosol, you're attaching them to the lactate ion, and so they're coming out of solution. So if your measurement of pH is the number of hydrogen ions in solution, this reaction would reduce the number of hydrogen ions in solution and increase pH. So some people might be looking into their, and I've already got an email about this, into their little 400-word assignment and wondering what they can talk about. Well, this would be probably one option that you can look up. And this source down here, Roberge, it's the guy's name is Robert Roberge. And in 2004, he wrote a paper in the American Journal of Physiology, which is a really short paper. It's about 2 pages, but it tells you exactly how this biochemistry works and why our textbooks are wrong. So one option is to look into this if you like. So I'm just going to check again the audio on the webcast to see if it's working. So just be a brief second. That's all. Okay. Now that we're back, you'll notice at the bottom of this screen okay, I already have a question, so I'll answer this. So is the accumulation of hydrogen ions what everyone confuses with lactic acid buildup? Yeah, basically that's the idea. The real kind of difference is that the actual hydrogen ions are coming from the breakdown of ATP and earlier enzymatic reactions that happen in glycolysis upstream of lactate production. And so, yeah, I think what's happened is that the whole idea of lactic acid, which is what you'll find in fermentation, has just been straight transferred over to human physiology or even muscle physiology without actually thinking it through and actually looking at the fact that lactic acid is not actually in existence. So it isn't dissociated. It isn't like you get production of pyruvic acid that then is converted to lactic acid, and then the hydrogen ion leaves. Pyruvate is already in solution. Pyruvic acid is already in solution, and it is a dissociated set of ions already because it is in solution. So that's well explained, I think, in the Roberge article. I'll try and put that actually on Moodle pretty soon, just so that it's a little easier for you guys to find. So with that said, I'm going to just finish off this slide. So the accumulation of lactate, in theory, could actually slow down glycolysis through what we call product inhibition. So if you did happen to have no way of getting lactate out of the muscle, then I guess what you could have is that the pyruvate could be converted into lactate, but then you could have accumulation of lactate. And what generally is believed is that this doesn't happen much because you have these what we call MCTs that you see on the screen, so monocarboxylate transporters, and I'll maybe spell that for you when I get back. These monocarboxylate transporters are very highly expressed in the cell membrane of muscle, and what they do is they move lactate out of the muscle as soon as it's produced. And you'll find that fast twitch muscle fibers have really high expression of this particular transporter in the membrane. And so they can move lactate out of cells like muscle cells very quickly. Interestingly, you have different transporters, MCT4 transporters, in other tissues in your body. So tissues like your brain actually have transporters that uptake lactate and then use lactate in their Krebs cycle, which is pretty cool. So they're able to convert it back into pyruvate and then allow that pyruvate to be streamed into the Krebs cycle and used for ATP generation, which is really neat. So you could have product inhibition, which could lead to, technically, to some fatiguing processes, but this hasn't been really well developed. So I'm going to hark a little bit more about lactate formation and that it isn't a poison. It's a key end product in glycolysis produced during glycolysis, anaerobic glycolysis. The rate of production increases as metabolic rate increases. So as you need more and more ATP, the amount of lactate that gets produced is higher and higher, hence the correlation between the two. But as I've mentioned, it is used by different muscles. So when you have a fast twitch muscle fiber producing lots of lactate, there are at least a few studies that show that there may even be transporters that move lactate from a fast twitch muscle fiber directly into a slow twitch muscle fiber where the lactate is converted back into pyruvate, which is pretty cool. And then other tissues, like I said, your brain uses lactate. And some studies that I know about, I wasn't at Brunel when they were being done, but they basically looked at the uptake of lactate in the brain by putting cannula or catheters at the base of the brain in some veins there, and also measured lactate in the arteries. In other words, we could look at essentially how much lactate was going into the brain in the artery and how much lactate was being consumed by the brain by taking the difference between those two blood samples, which is pretty cool. So they've shown, some colleagues of mine at Brunel were able to show that the brain uses a lot of lactate actually as a fuel. The heart also uses lactate as a fuel, which is pretty cool. So it's exported out of the muscle by transporters along with hydrogen ions. So at the same time as you are transporting lactate, this transporter specifically, the MCT, also moves hydrogen ions. And a key thing that's interesting is that you always have the production of lactate, which suggests at all times there is some basal amount of anaerobic glycolysis happening in pretty much every tissue. So nothing's kind of black and white. You don't use ATP only from the phosphagen system and ATP only from your Krebs cycle or ATP only coming from glycolysis. At all times, it's a bit of a mix of all three. It just depends on how long you've been contracting a muscle or how hard a tissue is working. That determines which one is going to provide the bulk of the ATP over that period. So the most important thing that happens due to the lactate dehydrogenase enzyme is what I've mentioned already. What you get is pyruvate producing NADH. The NADH gets consumed in the lactate dehydrogenase enzyme reaction. And the end result of that reaction is NAD, which can go back up to glycolysis and help with throughput or ensure that the flux through the glycolysis, the anaerobic glycolysis pathway, is as quick as possible. And you can even see the stoichiometric information here. A pyruvate molecule is C3H3O3, whereas a lactate molecule is C3H5O3. So you've got two hydrogen ions added to this, to a pyruvate molecule in order to produce a lactate molecule. Now, and the key thing here to note is if all NAD plus is used up, then glycolysis would stop. So in human cells, there's always another option though. You can always recycle or resynthesize NAD through other options. And that other option is what we call oxidative phosphorylation, which is essentially the Krebs cycle. So your overall reactions here, as you can see, glucose produces 2-pyruvate, 2-NADH, 2-ATP, and 2 hydrogen ions, whereas glycogen produces an extra ATP and actually consumes a hydrogen ion because you don't actually get it coming from the breakdown of that extra ATP. So some important aspects that you should consider. So muscle intermediate energy sources involve phosphagens, so ATP, ADP, and creatine phosphate. You replenish this system usually through aerobically produced ATP. So if you want to replenish the creatine phosphate, you have to wait for the Krebs cycle to provide that ATP. There essentially is no pyruvic acid in existence in solution, and thus lactic acid is not produced as the end product of glycolysis. Because the pyruvic acid is already dissociated into pyruvate and its hydrogen ion, lactate is produced, and actually hydrogen ions are consumed. And muscle glycogen is the preferential glucose source when metabolic rate is high because it gets that extra ATP. So we'll go through the aerobic glycolysis pathway, and again, the key features of the anaerobic glycolysis pathway are the continuation of glycolysis leading from the pyruvate molecule. So important aspects to focus on over the next few slides is entry of pyruvate into the Krebs cycle, because this seems to be a rate limiting step in order to produce ATP using oxygen. So that pyruvate dehydrogenase enzyme requires some time for it to get activated and is kind of the rate limiting step. And then the next step is that oxidation reduction reactions drive ATP synthase in the mitochondrial membrane. So you get the movement essentially of these electrons that drive the movement of protons across a membrane, which then create a concentration gradient in the hydrogen ion concentration on two sides of a membrane. These hydrogen ions then move through the ATP synthase, and during this process, they catalyze the reaction where ADP and inorganic phosphate are bound and essentially create your ATP. Now the muscle uses generally pyruvate when abundant and alternatively free fatty acids. So for the most part, whenever you have a lot of glycogen or a lot of glucose in your blood, your muscles will generally use glucose. But anytime that your blood glucose starts to drop, let's say when I go to bed, which right now it's about midnight, so I'll be going to bed in about a half an hour when I'm finished this, over the course of the night, I'm going to switch fuels from glucose or pyruvate to free fatty acids because they're more available. So the entry point again is this pyruvate dehydrogenase enzyme. And so a pyruvate combines with an acetyl-CoA, and this combining this synthesis reaction creates acetyl-CoA and a bit of CO2. It also creates one of the first NADH that you'll get out of the Krebs cycle. So if the activity of the PDH enzyme is not high enough, what would happen? I guess you guys can answer that maybe in the little live chat, and I can go over that in a minute. And the big question is, how do you increase the rate or the activity level of the pyruvate dehydrogenase enzyme? Now this slide's fairly busy. What you see up at the top is your acetyl-CoA. entering the Krebs cycle, going through a series of reactions, and each one of these reactions either kind of modifies the structure of the original acetyl-CoA. So the first thing that you get is oxaloacetate, which is a 4-carbon molecule combining with acetyl-CoA. The CoA is released from the molecule and you get a 6-carbon citrate molecule. So just to go back a second, just notice that this pyruvate dehydrogenase reaction is the first step where you actually get a removal of a CO2. So you had a 3-carbon pyruvate molecule, the production of NADH, which then creates a 2-carbon acetyl-CoA molecule and 1 carbon dioxide molecule. So you've gone from so as I've said, 2 3-carbon pyruvate molecules. Now you've got 2 acetyl coenzyme A molecules and the production net of 2 carbon dioxides at this phase of this cycle. So moving on, now we have oxaloacetate combining with 2 carbons, so a 4-carbon and a 2-carbon molecule, acetyl-CoA, the release of CoA, and then this next step is now a 6-carbon molecule called citrate. It's reorganized into isocitrate, and then the subsequent step when you get to alpha-ketoglutarate is the release of another carbon. So you're going from 6 carbons here at isocitrate to a 5-carbon alpha-ketoglutarate molecule. You also get the production of another molecule of NADH and some hydrogen ions. And you'll notice in the middle of this they refer to oxidative phosphorylation, and that's where these NADHs are going to go to the electron transport chain in order to donate their electrons and pump hydrogens across a membrane. Moving on from alpha-ketoglutarate, you have another reaction which produces another carbon dioxide molecule, another NADH, and you end up with succinyl coenzyme A. You get some what we call substrate level phosphorylation in the next step where you're producing succinate. In other words, a GDP, which is similar to an ADP, combines with an inorganic phosphate to produce GTP, which quickly donates this inorganic phosphate to an ADP molecule, producing 1 ATP in the Krebs cycle each time you go around the full cycle. So at this point you have succinate, which is a 4-carbon molecule. The next reaction to fumarate produces another electron acceptor called FAD-H2 from FAD, which will then enter oxidative phosphorylation. Fumarate undergoes another kind of reorganization with the addition of water, and then finally donates some more electrons to NAD, and producing more NADH for oxidative phosphorylation, at which point it becomes oxaloacetate, which is that 4-carbon molecule that goes through the cycle again. So these are called Krebs cycle intermediates. And if you have 2 acetyl-CoAs entering per glucose molecule, you'll go around this whole Krebs cycle 2 times for every glucose molecule that's completely oxidated in skeletal muscle. So if you go around this whole process twice, you will have a whole lot of NAD produced. You'll have at least 4 carbon dioxide molecules produced. You'll have at least 2 ATPs produced and at least 2 FADH molecules produced through this whole process. Okay, so let's go back for a second. We're just going to go back to this question that I had on the previous slide. So if the activity of the PDH enzyme is not high enough, what would happen? If the PDH enzyme isn't fully activated, you'll get accumulation of pyruvate, which would push pyruvate to be converted into lactate, and then lactate would be removed from the muscle. So that's the main thing that happens if PDH enzyme isn't at a high enough activity level. The way it's actually increased really depends on what you have accumulating in the muscle. So during contraction, you'll get a few products that actually activate the enzyme. So when you contract a skeletal muscle, you'll get an increase in ADP being produced. That ADP will actually activate the pyruvate dehydrogenase enzyme. And there are other molecules that allosterically modify the pyruvate dehydrogenase enzyme. And they're related to any time you see demand in ATP go up. I think another one might be NAD. So if you have a lot of NAD being produced, it causes the pyruvate dehydrogenase enzyme also to increase in activity. Thanks, Naya. It's good that somebody's actually going to try. And it's good that you guys actually are still getting what I'm putting out there. So it's great. So the key aspects of the Krebs cycle, you get a whole host of products, and the net reaction at the bottom is really what matters. So for each acetyl-CoA that goes into the Krebs cycle, you're going to consume 3 NADHs, an FAD plus, a GDP, an inorganic phosphate, and 2 water molecules. What you end up producing is 2 CO2, 3 NADH, 3 hydrogen ions, an FADH2, which is an electron acceptor, and a GTP, which you could substitute for an ATP, which you can see in the third row of this particular slide. Now that's one acetyl-CoA molecule. From every glucose molecule, you'll have 2 acetyl-CoAs, and so you'll get twice as many, say, NADHs produced, so you'll get 6 hydrogen ions, 2 FADs, and 2 ATPs being produced in the Krebs cycle. Now the next thing that happens is that the electrons that came off of various molecules in the Krebs cycle were taken up by NADH. And what they do then is find their way to the mitochondrial inner membrane. So you'll notice in this schematic you've got the outer membrane, which would be adjacent to the cytosol. You've got an intermembrane space, and then you have the inner mitochondrial membrane, and within that membrane you have a whole host of proteins that can pump hydrogen ions across from the inner matrix into the intermembrane space. Every time you provide some electrons, they activate essentially these 4 complexes and help with the movement of these hydrogen ions. So the NADH that came from the Krebs cycle find their way, probably through simple diffusion, to the inner mitochondrial membrane and one of the complexes of the electron transport chain. They donate those electrons to this first complex, which pumps a number of hydrogens across the membrane to increase the concentration of protons within the intermembrane space. Those electrons are then transferred over to another complex within the membrane, and then transferred a second time to another complex, which pumps more hydrogen ions across. And then finally, these electrons make their way to the last complex, help with the pushing or the movement of more hydrogen ions to create an even greater concentration gradient between the intermembrane space and the matrix of the mitochondria. This last stage, what happens is half an oxygen molecule, or what's known as molecular, or is atomic oxygen essentially, combines with some hydrogen ions and the electrons that were first donated at complex 1. These all combine to create a little bit of water, because your oxygen is your electron receptor within the body. So this is essentially where the oxygen is being consumed in the body or used up. So when you breathe in, ultimately the oxygen needs to get to this place where it can be used as the electron acceptor. And once it's accepted some electrons, it'll join with 2 hydrogen ions to produce a molecule of water, which is pretty cool. So finally, you've created this large concentration gradient in hydrogen ions between this intermembrane space and the inner mitochondrial matrix. The last item here is the ATP synthase, which essentially means that it's going to synthesize ATP. So as these hydrogen ions move down their concentration gradient from the intermembrane space to the matrix of the mitochondria, it catalyzes essentially the reaction which converts ADP, an inorganic phosphate, into ATP. So if we want to look at the whole process and all the products from the breakdown of glucose, we get this summary chart. And what you have here is a glucose molecule being broken down in anaerobic glycolysis to produce net of 2 ATP, a net of 2 NADHs, and some water molecules, and 2 pyruvate. Those pyruvate then can enter the Krebs cycle, where eventually you get the production of CO2. So if you have 2 3-carbon pyruvates entering the Krebs cycle, that means you'll have 6 carbons entering the Krebs cycle. And you'll see these carbons being released as CO2 at 2 points. 1 at the pyruvate dehydrogenase enzyme, and then 2 somewhere within the Krebs cycle. So you get a production of 6 total carbon dioxides, which you'll see down here in the summary equation. The other important aspect is that you get a couple of ATP being produced in what we call substrate level phosphorylation within the Krebs cycle. 2 more NADHs being produced at the pyruvate dehydrogenase enzyme, and then 6 NADHs being produced from within the Krebs cycle, and 2 FADs being produced from within the Krebs cycle. These donate their electrons to the various cytochromes or complexes within the mitochondrial inner membrane, which produce ATP at ATP synthase. And in total, you'll notice it says roughly 30 to 34 ATPs are produced. They get this number essentially by multiplying the theoretical value or theoretical number of ATP that can be produced from each set of electrons that are donated to your cytochromes. So in this case, you've got 2 for 10 total NADH, which each donate their electrons at the first complex, and theoretically each of these produces roughly 3 ATP each. So each NADH results in 3 of these ATPs, which you can see here. So if you enter in the first part, for each molecule of NADH, it's theorized that you get about 3 ATP being produced. Each FADH2 molecule enters the cytochromes or donates its electrons a little further down to one of the other complexes, complex 2, and that results in only around 2 ATPs, or maybe even theoretically less than 2 ATPs being produced per FAD molecule. Now, in biochemistry, you guys have probably heard that actually the amount is about 2.5 per NAD and 1.5 per FADH. This is definitely another issue with this particular textbook. It might be the same on the open textbook. So another good example of what you can suggest should be improved in the open textbook, as well as this one, providing some references and some information about where those are coming from. So the net result that you can see down at the bottom is one glucose molecule, C6H12O6, requires about 6 oxygen molecules in order for it to be completely broken down to produce 34 to 38 ATP. And the other key factor is 6 carbon dioxides are being produced. So you have an even ratio of oxygen used to carbon dioxide produced. Now the next type of fuel that you can use to produce ATP is a free fatty acid. So if we think a little bit back, you've probably heard of a triglyceride. A triglyceride essentially is glycerol, which has 3 different free fatty acids, or 3 of the same free fatty acids attached to it. So within your adipose tissue, which we all have, fat cells essentially store free fatty acids in what we call triglycerides. When you need fuel, in other words, you're sleeping, you've used up most of your glucose and you're trying to save it for brain function while you're asleep, you'll also you'll start to release triglycerides from your adipose tissue. Those triglycerides will travel to, let's say, a skeletal muscle. The free fatty acids will be released and they'll traverse the membrane and enter that muscle and become one of the fuels that it uses. So at the top of this particular slide, and probably the last slide we'll see today, you have a free fatty acid. In this case it says C18, meaning an 18 carbon fatty acid. And the full name is at the top there. Now, this process is called beta oxidation, and essentially what it means is that this free fatty acid, this 18 carbon free fatty acid, can be cleaved every 2 carbons, and each of these pairs of carbons can be entered into the Krebs cycle. In order to do this, you need to consume some ATP, as you can see here. But you also get the donation of electrons to FAD and NADH, which can then enter oxidative phosphorylation, just like you saw previously. So at the end of this reaction, you cleave off, as you can see here, 2 carbons, and they combine with a CoA or a coenzyme A to produce acetyl coenzyme A. The remaining 16 carbons go back up to an earlier phase of beta oxidation, create more FAD, create more NADH, and then as they move through the system again, another 2 carbons will be cleaved off, and now you'll have a 12 carbon molecule, free fatty acid that's 12 carbons long, be recycled back up to the start, and go through the process. So this will, on an 18 carbon free fatty acid, you're going to go through this process 9 times and produce 9 acetyl-CoA molecules that will enter the Krebs cycle. So those acetyl-CoA enzymes go through the Krebs cycle, 9 of them. They produce 9 ATP. And they also produce the other coenzymes, or NADHs, that you would normally see with each acetyl-CoA that you saw in previous slides. So the net result after you break down an 18 carbon free fatty acid is the production of 139 ATP per molecule. So I'm going to leave it actually there for today. If you have any questions, I'm going to try and look at them and just type them in here while you have another 5 minutes. And then what we'll do is tomorrow I will be back online. I'll probably come online a bit early for you guys to ask questions, as well as if you have any concerns. The other thing I'm going to do is try and send an email with times when I'm available via Skype, or even just available through this on later days this week. So hopefully this isn't being too difficult. I think this will be posted in a little while, which will be useful. And if I notice anything that needs to be corrected, I'll mention it in the comments or within Moodle in the next couple of days.